Good morning, students. In today's lecture, I will be discussing about another set of genes which are shared by the protostomes as well as by the deuterostomes. As in the previous lectures, you have learned that there are Hox genes. They are the cluster of genes present on chromosomes which are responsible in determining the structural pattern or structural organization of the living organism, be it a small organism as small as hydra and big organism as big as elephant or giraffe. So this Hox gene is present in simpler organism, invertebrates to vertebrates, or we can say that they are present in both deuterostomes and protostomes. Similarly, like Hox genes, there are Pax gene, PAX genes. These are also set of genes which are responsible for the formation of eye in invertebrates as well as vertebrates. Then we have the Tin Man gene. The mammalian counterpart of it is called NKX2, NKX3, NKX4, NKX5. This is a counterpart to Tin Man gene, and they are responsible for the formation of heart. Though the structure of heart in invertebrates may differ from the structure of heart in higher animals, but the main function of the heart is to uh, pump the fluids, be it hemolymph or blood, through the circulatory system of the animal or through the uh, into the body of organism. So besides these, there is another set of genes which are shared by the protostomes as well as deuterostomes, and these are the genes responsible for the transcription factors which are involved in the head formation. In Drosophila, the brain is composed of three segments called the neuromeres. Neuromeres are morphologically or molecularly defined transient segment of early developing brain. Rhombomeres are such segment that make up the rhombocephalon or the hindbrain. More controversially, some argue that there exist early developmental segments that give rise to structure of the midbrain, that is mesomere, and forebrain, prosomeres. Neuromeres are the segments of neural tube that establish the embryonic brain during development. They can then be divided up so that each is carrying different and unique genetic trait expressing different features in development. As I have already stated that in Drosophila the brain is composed of three segments called neuromeres, and these neuromeres are specified by three transcription factors. Genes encoding these factors are called the tailless and orthodentical, which are expressed predominantly in the anteriormost neuromere, and empty spiracle, which is expressed in the posterior two neuromeres. Loss of function or mutation of the orthodentical eliminate the anteriormost neuromere of developing Drosophila embryo, and loss of function or mutation of empty spiracle eliminate the second and the third neuromeres. Similarly, in frogs and mice, the homologue of genes are OTX1, OTX2, EMX1, EMX2. These are the counterpart or homologues for the head genes present in the Drosophila. So these genes are also expressed in the brain in frog and mice. Though the exact pattern of transcription are not identical, the OTX2 gene has been experimentally knocked out, and the resulting mice have neural and mesodermal head deficiencies. In humans, the mutation of EMX2 lead to a rare condition known as schizencephaly, in which there are clefts ripping through the entire cerebral cortex. Even though the Drosophila orthodentical and empty spiracle genes are specified by the bicoid and hunchback gradient in the mammalian, OTX and EMX genes are induced by anterior dorsal, mesoderm and endoderm. So the nutshell of what I have spoken is that there are set of genes which are present not only in invertebrates but are also present in vertebrates, and these genes are homeotic genes, that is the conserved genes which are found in lower animal to higher animal, and they regulate the pattern of head formation and the pattern of nervous system formation. So we can say that the ancestor of all bilaterian organism had sensory organ based on Parkes 6, head based on orthodontal, dentical, empty spiracle and tailless gene, heart based on Tinman, and the anterior posterior polarity based on the expression of Hox gene.